Good morning, all. I am Srishna K. First MSc Biology, St. Mary's College, Thrissur. Today we are dealing with the topic industrial enzymes, vitamin B12, and single cell protein. First of all, industrial enzymes. We know enzymes are biomolecules that catalyze chemical reaction. Nearly all enzymes are proteins. In enzymatic reaction, the molecules at the beginning of the process are called substrates. Enzymes converts them to different molecules called products. Industrial enzymes are those which are used for industrial production and also these are produced in industry. Example, proteases, lactases, amylases, etc. Amylases. Starch. A glucose polymer is one of the most widely available plant polysaccharide. Amylases are enzymes that hydrolyze starch. One of its main use is in the production of sweeteners for the food industry. The hydrolysis of starch with amylase first result in the production of short chain polymers called dextrins, then the disaccharide maltose, and finally glucose. The most important enzymes in the starch saccharification process are alpha amylases, beta amylases, glucoamylases, glucoisomerase, pulleinases, isoamylases, etc. Alpha amylases. These are extracellular enzymes which hydrolyze alpha 1,4 glycosidic bonds. These are endoenzymes splitting, uh, splitting the substrates in the interior of the molecule. These are formed by many bacteria and fungi. Bacteria which produce alpha amylase are Bacillus subtilis, Bacillus cereus, Lactobacillus, Micrococcus, Pseudomonas, Arthrobacter, Escherichia, etc. Some alpha amylase producing fungi are from the genera Aspergillus, Penicillium, Cephalosporium, Mucor, Candida, Neurospora, and Rhizopus. The mole weight of various alpha amylases do not differ considerably. They all contain a large proportion of tyrosine and tryptophan in enzyme protein and most require calcium as a stabilizer. Most important amylases are produced by Bacillus amyloliquefaciens, Bacillus licheniformis, and Aspergillus oryzae. Amylases are much more extensively used than Aspergillus. Beta amylases. These are usually of plant origin. Some microbial producers are also known. They are Bacillus polymixa, Bacillus cereus, Bacillus megatherium, Streptomyces species, Pseudomonas species, Rhizopus japonicus, etc. Bacterial beta amylases have greater heat resistance than plant beta amylases and pH optimum is also higher. In contrast to alpha amylases, calcium is not necessary for stabilization and activation of bacterial beta amylases. Glucoamylases. It acts on starch by splitting glucose units from the non-reducing end. Microorganisms used to produce glucoamylases are Aspergillus niger, Aspergillus oryza, etc. Today, industrial fermentation of glucoamylases is carried out almost exclusively in submerged fermenters up to 150 meter cube in volume. Due to increase in demand for glucoamylases in fructose syrup production, smaller systems which involve growth on the surface of liquid or solid are no longer economical. Glucose isomerases. It causes isomerization of glucose to fructose. The reaction is reversible and mixture of glucose and fructose produced. The enzyme has become of commercial value because the price of sugar has increased compared to that of starch. Starch can be converted to glucose by either acid or enzyme hydrolysis. The glucose isomerase used must fulfill the following criteria: low pH, optimum to avoid side reactions, high specific activity, high temperature optimum. Most important glucose isomerase producers are Bacillus coagulans, Streptomyces rubiginosus, etc. L-asparaginase, used as an anti-tumor agent in the treatment of some leukemia and lymphomas. The principle behind the use of this enzyme as an anti-tumor agent is as follows. Metabolism of these tumor cells require L-asparagine, and since asparagine is split into aspartic acid and ammonium ions by asparaginase, these malignant cells can be destroyed. The enzyme is produced by several bacteria, but for commercial production, only mutants of E. coli, ATCC 9637, Serratia marcescens, ATCC 60, and Erwinia carotovora are used. At the end of fermentation process, the cells are broken up and the enzyme is crystallized. Proteases. After amylases, the second most important industrial enzymes currently are proteases. About 500 tons of this enzyme is produced per year. Primarily, proteases are used in detergent industry and in the dairy industry. Other areas in which proteases are used include pharmaceutical industry and the leather industry, food industry, film industry, and waste processing. Proteases are produced commercially both from bacteria and fungi. Proteases on market include alkaline, neutral, and acid proteases. Alkaline proteases. Many bacteria and fungi excrete alkaline proteases. Most important producers are Bacillus sorry Bacillus strains such as Bacillus firmus, Bacillus licheniformis, Bacillus amyloliquefaciens, etc. Streptomyces strains such as Streptomyces fradii, Streptomyces griseus, Streptomyces rectus. Fungi Aspergillus niger, Aspergillus zoae, Aspergillus oryzae, Aspergillus flavus. Enzymes used in detergent are mainly proteases from Bacillus strains. Alkaline proteases have many features of value for use as detergent enzyme. Stability at high temperature, stability in alkaline range, pH ranging from 9 to 11, stability in association with chelating agents and perborates. Neutral proteases. It is excreted by both bacteria and fungi. Producing organisms include Bacillus subtilis, Bacillus cereus, Bacillus megaterium, Pseudomonas aeruginosa, Streptomyces griseus, Aspergillus oryzae, Aspergillus zoae, etc. These are relatively unstable, and calcium, sodium chloride must be added for maximal stability. The pH of activity is fairly narrow and this enzyme not very stable to increase temperature. It is quickly inactivated by alkaline proteases. Because of these limitations, they have restricted industrial applications but do find some uses in leather industry and in food industry. Acid proteases. It is in this category are rennin like proteases from fungi which are chiefly used in cheese production. In addition, other acid proteases which are similar to mammalian pepsin are also on the market. These enzymes have pH optimum of 2 to 4. These are used in medicine in digestion of soy protein for soy sauce production and to break down wheat gluten in the baking industry. Rennin. It is milk coagulating enzyme used for cheese production. Rennet has been used for generations as milk coagulating enzyme. It is an extract from fourth stomach of 3 to 4 week old calves which have been raised on milk. The purified enzyme is called rennin, chymase or chymosin. A variety of fungi and bacteria have been isolated as rennin producers. Most important genera of bacteria are Bacillus, Corynebacterium, Lactobacillus, Pseudomonas, etc. Most important genera of fungi include Aspergillus, Candida, Endothea, etc. Enzyme system must meet the following requirements: good coagulation of casein, good odor and structure of cheese, no unpleasant of its own, non-toxic, low proteolysis in order to prevent the development of bitterness in the ripening process, low lipase activity to avoid the development of rancidity in the cheese. Pectinases. Pectinases preparation contains at least 6 enzymes. splitting pectins at different sites of the molecule. Basic structure of pectin is alpha 1,4 linked galacturonic acid with up to 95% of its carboxylic group esterified with methanol. Pectinases are classified according to their attack on pectin molecule. A number of commercial fish produce fungal pectinases using Aspergillus niger or Aspergillus venti. Both surface and submerged processes are used. Pectinases have been discovered in other fungi and also in bacteria, protozoa, insects, and higher plants. Lipases. Lipases splits fats into monoglycerides and fatty acids. They are usually extracellular enzymes. Fungi producing lipases include Aspergillus, Mucor, Rhizopus, Penicillium, etc. Yeast producing lipases include Tor- Torulopo, sorry, Torulopsis and Candida. Bacteria producing lipases include Pseudomonas, Staphylococcus, etc. Of these organisms, only Aspergillus, Mucor, Rhizopus, and Candida are used commercially. Commercial, commercial use of lipases is limited. They are primarily marketed for therapeutic purpose as digestive enzyme to supplement pancreatic lipases. It is also found in some dairy industry. Lactases. It is also called beta galactosidase. It splits lactose into glucose and galactose. The enzyme is intracellular in bacteria and yeast, but it is secreted by many fungi. The enzyme produced commercially are obtained from fungi or yeast and are less well known. Lactase of commercial interest include those from fungi, Aspergillus oryzae and Aspergillus niger. Those from the yeast, uh, Cluiver, Cluiveromyceae lactis, Cluiveromyceae fragilis, and Torula cremoris, and Bacillus species. These are used as digestive enzyme in case of lactose intolerance and as a feed additive to increase the nutritive value of some animal feeds. In dairy industry, lactases are used to break down lactose in milk. Penicillin acylases. Many penicillin antibiotics used in medicine are produced semi-synthetically by chemical modification of basic penicillin ring structure. The starting material for chemical modification is non-acylated thiazolidine beta-lactam ring system of penicillins, 6-amino-penicillinic acid. 6-APA is excreted in low concentration by penicillin chrysogenum when the organism is grown in the medium without addition of phenyl acetic acid as a precursor. The process for production of basic ring system can currently be commercially utilized due to its high cost. 6-APA can also be produced by splitting of acyl side chains of microbially produced penicillin G with the enzyme penicillin acylase. Penicillin acylase thus have commercial use in production of semi-synthetic penicillin. Advantages of industrial enzymes. Hazards-free and environment-friendly production is possible. Purity of the product can be ensured. Production cost is low. Simple nutritional value. Cheap carbon and energy source. Easy genetically manipulation. Easy product recovery. Disadvantages of industrial enzymes. Sometimes product stability is not good. Risk of pathogenicity. Chances of contamination. Low efficiency. Require additional measures such as sterilization. Our next topic is vitamin B12. Vitamin B12 is also known as cyanocobalamin. Is synthesized in nature by microorganisms. It is required by animals and it is present in every animal tissue in very low concentration. Although the substance is isolated from tissue is cyanocobalamin in the cell, only the coenzyme form is present. Cyanocobalamin is not appearing until the product recovery stage. The vitamin B12 needs of animal are covered by food intake or by absorption of it produced by intestinal microorganisms. However, humans obtain vitamin B12 only from food, since B12 synthesized by microorganisms in large intestinal tract can't be assimilated. Its sources are dietary intake is exclusively from animal sources, example milk, meat, egg, etc. Although some bacteria make vitamin B12, it is probably not in a form that can be used by the body. Fortified breakfast cereals can be useful source of vitamin B12. Function, normal cell division and normal blood formation for the normal structure and function of nerves. Together with folate and vitamin B6, it is required for the maintenance of normal blood homocysteine levels. Raised blood homocysteine is a risk factor for cardiovascular disease. Deficiency. Dietary deficiency is rare, although it is sometimes seen in vegans who obtain virtually no vitamin B12 in their diet. Deficiency is also caused by a lack of intrinsic factor, the substances needed for absorption of vitamin B12. This leads to a type of pernicious anemia, in which red cells are enlarged, and to neurological damage, paralysis. The structure of vitamin B12. And our final topic is single cell protein. Single cell protein refers to the crude, a refined or edible protein extracted from pure microbial cultures, dead or dried cell biomass. They can be used as a protein supplement for both humans and animals. Microorganisms like algae, fungi, yeast, and bacteria have very high protein content in their biomass. These microbes can be grown using inexpensive substrates like agricultural waste, wood shavings, sawdust, corn cobs, etc., and even from human and animal waste. Microorganisms used for the production of single cell proteins, algae, spirulina. It contains 65 to 70% of protein. Chlorella pyrenoidesa, good source of vitamins, minerals, and antioxidants. Fungi, Agaricus campestris and Saccharomyces cerevisiae are the two fungi extensively used for SCP production. Mushrooms are rich in protein and antioxidants and directly consumed by human. Bacteria, Pseudomonas fluorescens and Lactobacillus widely used as SCP. They have high protein content. Production of single cell protein. The production is carried out in the following steps. First, selection of suitable strain, fermentation, harvesting, post harvest treatment, and finally processing of SCP for food. The first step is selection of suitable strain. Selection of a suitable microbial strain for SCP production must take several characteristics into account, including growth rate, productivity, preferably low cost substrates to be used, temperature and pH tolerance, oxygen requirements, heat generation during fermentation and foaming characteristics, growth morphology and genetic stability in the fermentation, ease of recovery of SCP and requirements for further downstream processing, safety and accessibility. The second step is fermentation. Fermentation is done in a large chamber either of glass or stainless steel called fermenter. Fermentation should be done under sterilized conditions. Usually, fed batch cultures are used for the fermentation. Controlled conditions are necessary such as temperature, pressure, pH, humidity. Harvesting. When the colonies of microbes are fully developed, they are then harvested. The bulk of cells are removed from the fermenter by decantation. Fourth step is post-harvest treatment. After harvesting, the cells are subjected to a variety of processes. Post-harvesting treatments include steps like separation by centrifugation, washing, drying, etc. And the final step is processing of single cell protein. Pure protein isolation can be done by disrupting the cell wall through crushing, crumbling, cycles of freezing, thawing, grinding, and thermal shocks. Uses of SCP. It is rich source of protein. 60 to 70 percentage. Vitamins, it's a rich source of vitamins, amino acids, minerals, and root fibers. It is a popular health food. It provides valuable protein rich supplement in human diet. It lowers blood sugar level of diabetes. Provides instant energy. It is extremely good for healthy eyes and skin. Provides the best protein supplemented food for undernourished children. Used in therapeutic and natural medicines. Controlling obesity, control lowers blood sugar level in diabetic persons, reducing body weight, cholesterol and stress, prevents accumulation of cholesterol in the body, used in cosmetics products for maintaining healthy hair, production of different herbal beauty products like bio lipstick, herbal face creams, etc. In cosmetics, important role in maintaining healthy hair, many herbal beauty products used in poultry as it serves as an excellent and convenient source of proteins and other nutrients. It is widely used for feeding cattle, birds, fishes, etc. Advantages of SCP. Large scale single cell protein production has multiple advantages over conventional food production practices, such as microorganisms have a high rate of multiplication, which means a large quantity of biomass can be produced in a comparatively shorter duration. The microbes can be used easily genetically modified to vary the amino acid composition. A broad variety of raw materials, including waste materials, can be used as substrate. This also helps in decreasing the number of pollutants. Production is independent of climatic conditions. Disadvantages of SCP. In spite of many advantages, there are few drawbacks. Single cell protein has not been widely accepted for human consumption owing to certain problems as follows. High level of nucleic acid in biomass makes it difficult for consumption as it may lead to gastrointestinal problems. The biomass may trigger an allergic reaction if the digestive system recognizes it as a foreign product. The presence of nucleic acid is high content leads to elevated levels of uric acid. In certain cases, the development of kidney stone and gout if consumed in high quality. Possibility of the presence of secondary toxic metabolites which results in hypersensitivity and other skin reactions. The capital cost of production is high as sophisticated machinery is required. Thank you.